Ok, tot arreglat, podem començar. Bon dia a tothom. It's a pleasure to be able to open this session, and I'm just going to give you a very, very broad and general introduction to what we are going to see. You all know the advertisement we did, and this is something organized by the European Academy, of which we have a hub in Barcelona, and the hub is being directed by me, and we have here Maite, that is the person that has had all the work of putting everything together. Thank you very much, Maite. The Academy, the Academia Europea is a big academia of Europe. You have here more or less a description, and the idea is to first to be a lobby of science in Europe, and for that we have the best scientists all over Europe, and it's a very good place of lobbying in the European Union and to other places, and helping to disseminate science into the population. And this is something that we do in the Barcelona hub, that we are part of it. This Barcelona hub, the first thing is that we are settled in the Fundació Catalana per la Recerca i la Innovació, of which we have Jordi, the director here, and we are very grateful to them, because this is the only way of existing for us, because the Fundació gives us the possibility of enhancing our work to do many other things. So, and they have a very nice premises just in front of the Arc de Triomphe, and this is our web, and we organize many different things. Usually the best known are the scientific dialogues, of which we do two levels of that. One we call Frontiers, in which we explore the limits of science and mainly the social implications of them. And the second is the Agora, in which we intend to be more close to the general population. Here are the two next of this meeting. The first one is on ecological collapse, and the second one, that will be in Catalan, on the sobremedicació. And in both cases, we have very good speakers that are going to put us the point. So you are invited to come to these activities. And today we have one of our activities that we intend to have a place, or to be active in the art and science world. And today, for us, it's a big pleasure, because we have been able to gather together some of the best people working on art, having this science view, and we are going to enjoy the talks we are going to have this morning. So you have all the introduction here, the program, sorry. Here we will have in this order. Oh, it's different, this one from the one I have in the table. I don't know whether Marta begins or Anna begins. It's different one from the other. You are the boss. First, Marta. Okay.
First, Marta. Okay.
Okay, sorry, I didn't know. And for doing all that, as I am a pure scientist having some art disgregation, and what we did is to contact Luis Nacenta. He is the person in the, let's say, the local person that has a broad vision of art and science, and we have been discussing many different things. So we thought that he would be the best person to put together a fantastic program, and he is the boss of all the session. What we have is here in this wonderful place. I hope that you will enjoy the setting. We have outside, there is a very nice room also, because all the clocks you are going to see outside, for many years it gave the official time in Barcelona and neighboring populations. And even now, walking in the Ramblas, when looking at outside of the building, you can see a banner saying official time. So the time that was put here was the time, so nothing else. And in the afternoon, very close to here, we'll have a hands-on, a workshop by both Marta de Menezes and Anna Dumitriu that will be extremely interesting. So I have nothing else to say, but I want to do a small advertisement that yesterday we opened the position of artists in residence in our department at the UPF Biomedical Department. Last year we had Jo Milne, that she is here. This year we have had Pep Vidal, and we have put these landscapes just for you, if you want to apply, or you know someone that wants to apply. So thank you very much for being here. I'm sure we'll enjoy, and Luis, the floor is yours.
Gracias, Jaume. Bon dia. Thank you, Jaume and Maite, for inviting me, for making this possible. It's always, I think, a privilege to have some time and the chance just to speak about the things that interest us, worry us, and that we love. When we thought of the title, that it's a bit, now I see that it's a bit convoluted, it's a bit complex, but ways of knowing, lines of contact between science and art, it's a bit too much. But the idea was ways of knowing. It was because I think all this is it's about knowledge in the end, and we should not just surrender to the idea that knowledge belongs only to the science space. And I think you all here know that or defend that, but that's a work that we still have to do. And it's very interesting that when you think of knowledge, it's not, I don't think we can really define such thing as an artistic knowledge or a scientific knowledge. It's just a common place. And then the lines of contact, that is because we now have, for some time now, I mean, quite strong institutional context for art and science. I mean, this is a thing that is very important. European programs that promote that. I mean, but still, we have to fight for this endeavor, not only to be something that a few of us do because we like, and something that becomes like a specific professional expertise. I think it's very important that it's something that affects deeply science in general, affects deeply art in general, and affects deeply society in general. And those lines of contacts are this idea that I am walking and Jaume is walking, we doing different things, but we come together for some time. We have a line that for some stretch we are walking together and we are in contact. We are not only just sharing ideas, but we are sharing experiences. So we try to construct, to build a knowledge that it's not only the possession of some ideas, but also the experience of knowing how to do, knowing how to be, knowing in the end how to live together. So let's make this journey today about details, about specificity of experiences. We know the frame. We know it's very important that science and art collaborate. Okay, that we know, but how we do it. So enough for the introduction because we are already late. So I will invite. So who's first in the end?
Anna.
Anna? Okay. Yes, because Giuseppe, now I understand what you were saying. Giuseppe has to go. Okay, so I will invite Anna and Giuseppe. I think you are there, Anna. Giuseppe, since, perhaps I don't know if you want to, perhaps you can sit later. Okay, yes, so you can relax. So the dynamic of the thing is a series of 4 conversations, but the conversations are not exactly symmetrical, but Anna Dumitriou will just do it's 20, 25, half an hour presentation. Yes? What's going on? Okay. Okay, I can talk for hours, don't worry. So the dynamic is that Anna will do a presentation of about 25, 30 minutes, whatever she feels like. But then we thought it was interesting to have, it's not a replica, it's not although that place really looks like a court of law. And it seems like, like we are judging art and science and someone is pro. It's not that. That's a conversation. That's a conversation. So we invited 4 relevant people living in Barcelona. I don't like that we are the locals. It's not that. Josep, Lua, Paula, and Joan, they are all very active in the international scene. Professionally, it's just that they happen to live in Barcelona. So it's beautiful that the locals, meaning people that live here and work here, we receive our guests today and we dialogue with them. But I don't like that idea of the locals. So that's the idea. We will listen to the presentation by Anna, and then we will have the conversation between Jaume, Josep, and Anna. And I will, as we wait for the computers to do the thing, I will present them both. So Anna Dumitriou, she works in bioart, sculpture, installation, and digital media. And the way you describe your work is to address the relation to our relation to infectious diseases, synthetic biology, and robotics. That is the way that Anna Dumitriou describes her work. Josep Parelló is a physicist, and he's been working for some time now in complex systems, in citizen science. He's been very active in our cultural context in those fields. She already knew Anna, so you already are aware of each other's works. I would like to mention that Josep is part of the group in Universitat de Barcelona called Open Systems. And I read the description. I want to read the description of what the Open Systems group does, because I think it's very relevant. So the description is that's a group working toward an interdisciplinary practice where art and citizen participation are a fundamental part of science. That is, I think, the best idea we can start with. It's not that, well, it's good that science is somehow related to art. Well, it's important that citizens somehow are there. No, that's not what the Open Systems group are saying. They're saying art and citizen participation are a fundamental part of science. So the alarm is alarmed by that statement. So if we are ready,
yeah. Can you hear me okay? Am I amplified to the right level? Sorry about the blip there. I had installed the PowerPoint on here, but they deleted it just before I started. So, yeah, I'm very pleased to be here. Thanks to everyone for inviting me and Yusuf for discussing with me. I thought I'd start off by talking about a project that we did here in Barcelona, just to sort of indicate how things work. I'm fascinated in infectious diseases, more than anyone probably that you'll ever meet. And I'm obsessed with things like tuberculosis, cholera, Yersinia pestis that causes plague, and all the kind of the diseases of history that are still very prevalent in a lot of global health settings. And we did a project about tuberculosis, which was based on the Picasso painting or drawing here that's in the Picasso Museum called Poor Geniuses, where it's it's just a bed where someone's dying of tuberculosis. And I've done a lot of work with Oxford University, the Modernising Medical Microbiology project, and the Cryptic project there, which is to do with mapping whole genome sequencing TB and all sorts of things around that. So this is a piece that I made. It's a little tiny piece of it. These lungs are little, actually. Marta has some, doesn't she, in your art collection? They're lungs made out of wool and dust, made in the images of diagrams that you would get in leaflets that they would give to patients that would explain how they would collapse your lung to enable it to fight off TB, tuberculosis. And it's impregnated with the extracted DNA of TB that I extracted in a category 3 lab at Oxford University. And I made a kind of replica bed, I guess, if you like, based on the Picasso bed. And it's here, it's got this little symbol on it, is the Crusade Against Tuberculosis, as it was called, by the American Lung Association. They used to sell these pins, and it's the 2-bar cross. So it's got interesting kind of colonial type issues with the whole symbol as well. And that's me adding some of the tuberculosis DNA to the bed. Tuberculosis has been very linked to creativity. So, you know, if you've got some scientists in the room and you're thinking, how can I be more creative? Then maybe you could infect yourself with tuberculosis. It's called the genius germ hypothesis, which is really interesting, linked to the poor geniuses idea. And I've made a whole series of works called hypersymbiotics, which are ways in which we could possibly use some of the features, shall we say, of bacteria to improve ourselves. Now, unfortunately, you would have to go through the symptoms of TB if you caught it. Well, there's sort of it's probably about a 10% chance that you would get sick from it, and of that, you might die if you didn't have any treatment. About half of you would die if you didn't have any treatment. But luckily, we're in the West, very modern medicine and things like that, so it's highly likely it would be whole genome sequence, and they'd know exactly what antibiotics to treat you with, and you could be treated. So you'd only really have the benefits. Now, you might ask, how can I catch TB, or can you supply it to me? I can't. I don't personally have it at the moment, as far as I know, anyway. But it tends to, in Europe, it tends to be something that's caught by people living chaotic lives, would be the medical term. So homeless people, drug addicts, and things like that. But anyway, I made these creativity pills. They're coated with gold because gold's obviously always good in medicine, isn't it? As we know from history. And they have the extracted DNA of tuberculosis in them. And that's a bigger view of my hypersymbiotics piece, the latest one, which is my post-pandemic edition, which was commissioned by it was commissioned by Medical Museum in Copenhagen and then exhibited by the Nobel Prize Museum in Stockholm. And that's got a whole load of different sort of, shall we say, drugs that can improve the world. We've got carbon capturing yeasts. There's a plasmid in there that can increased serotonin production. You can add it to a probiotic E. coli and have an overgrowth of serotonin. So you might be really, really happy if you had that. However, it's not been tested in humans, so we don't know, how much that E. coli would grow in your gut, and you might end up with an overdose of serotonin. So not yet recommended for human consumption. This is a piece I made that's called Coming Back. And it's got that 2-bar cross on the doorknob. The whole piece stemmed from that because I found that. It's a doorknob from an American TB sanatorium. So all the patients would have touched that doorknob. And the door's been carved with the images of TB, how it damages the lungs, and painted with those images. And then it's all impregnated with what's called the Euro-American strain of TB. And this strain of TB is the one that comes from Europe and comes from America and is the most prevalent strain in places like Bangladesh, India, Pakistan, where people try to think they could shut out the immigrants with the TB and stop it coming back. So that's why I use that phrase. But of course, it really is coming back because we took it over there in the first place with colonialism. So just to kind of tell that story through the work. This is my hand in a Category 3 hood at Porton Down, which is the highest security laboratory in the UK on a military base. And this is me holding a mixture of Staphylococcus aureus, which is an everyday bacteria. 30% of us probably carry it around, at least 30% of us carry it around in our nose. It likes to live at the front of your nose, where it can avoid your immune system when it heats up to kill it. But the other part, the little round colonies there, is Yersinia pestis, which causes plague, which you probably heard of. And if you ever are growing bacteria in the lab, because I'm going to teach you how to grow bacteria this afternoon yourself, if you grow these things, they're called umbonate colonies. So it's like that. It's like a fried egg, if you imagine it. If you grow something that looks like a fried egg, you might want to report yourself to the authorities because you might have accidentally cultured the plague. If you culture anthrax, it's grey, and if you touch it with a loop, like one of the laboratory loops, it will go into peaks like a very good meringue. You know when you cook meringue? So that's how to spot that if you ever accidentally culture it. And yeah, there are some pics of killed animals, definitely in Spain, I know for a fact. So it's always a possibility. This is my plague dress. This is when it was shown at Timisoara for the European Capital of Culture last year. And it's a 1665 dress design dyed with walnut, which Nicholas Culpeper said was probably a very good treatment for plague. Now, I'm not sure it is, so probably antibiotics would be better. But it's this beautiful kind of beigey colour, which there's notes from the period that say that it travelled on light-coloured cloth. They thought plague travelled on light-coloured cloth. And actually what they would do when somebody died of the plague, they would bundle up their linen after they died and then go and sell it. And I believe it was Spanish researchers actually who discovered that actually it's not just fleas, but a lot of it was human body lice that were spreading plague. And so they would live longer in these bundles of cloth that was wrapped up. And when people unwrapped it, they would inhale the bacteria and the feces in the bundles, and that's how they would get pneumonic plague, and that's how that was spread. And it's got lavender in it, which was held in these poses because they thought it was caused by bad air, miasmas, and the embroidery on the front's actual period embroidery from the time of the Great Plague of London, and that's impregnated with Yersinia pestis bacteria, the DNA of that I extracted at Porton Down, at Colindale in London. And that's how it was shown this year in the Thackeray Museum in Leeds, alongside another piece called Xenexton. This is Xenexton. Paracelsus, the historic physician, said that plague was such a big issue that that we should have an amulet to ward it off specifically. So before that, people just wore amulets, these sort of things around their neck, or carried something with them to ward off disease, just all disease. But it was such a big issue, he said, we need one specifically targeted. And I really like that, because it's a bit like when we talk about targeting antibiotics instead of using broad spectrum antibiotics that can destroy your gut microbiome or trigger more antibiotic resistance. So he said, let's do this. And he said, well, it should probably contain a toad. It should probably contain a toad, because we all know that if you carry a toad around, dried toad, that will ward off disease. And he said it should probably contain menstrual blood. Okay, because that's always really good for this sort of thing as well. And he said sapphires, or if you're poor, amber. So because we all know if you get a plague bubo, you can put a sapphire on it and it will turn black and leach away the disease. Not sure. But anyway, what I really love about it is over the next, he said it won't happen in my lifetime. The natural philosophers, the physicians of the future will discover this. And over the next 100 years, you get a debate with the scientists, physicians, let's call them. And they say, well, it should be paste of toad, because that'd be better. And it's very much like sort of how we do science now with like improving things, but it's kind of mad. Because then, no, powder the toad, powdered toad, that's the way to go. And menstrual blood won't do it. It needs to be the menstrual blood of a virgin. The first menstrual blood of a virgin, gathered on a full moon. So this necklace that I've made has all that. It's 3D printed. It's actually brass plated with gold. I couldn't afford to do gold 3D printing. They don't actually do it that big. It's that big. So we've got the menstrual blood, we've got the sapphires. They're actually lab sapphires. And you've got the full moon, the texture of the toad and things like that. But mine's the next, and obviously the walnut dyed silk there, like Culpeper said. But Misenexton will actually ward off disease. I don't know if there's another slide of it, but Misenexton will actually ward off disease because I did a collaboration with the scientist who's created from the Oxford Vaccine Group now at Surrey University, who's created the latest plague vaccine, Yersinia pestis vaccine, based on the ChAdOx1 platform. So it's the same platform that they made the Oxford AstraZeneca COVID vaccine from, but you can plug in different bacteria to it and use different vaccines. Also done a lot with storytelling. This piece, Ex Voto, collects people's stories of their experiences of taking antibiotics and working with antibiotics and their experiences of infectious diseases. And it's sort of talking about kind of making a wish or giving kind of thanks for the antibiotics, really. And so they're all made by lots of different people. I made some more last weekend. It's been shown all around the world. A slightly complicated project that I did involves kind of editing out antibiotic resistance. So this piece on the side there, I had to have a typhoid vaccination to make this. By the way, I trained in fine art. I've just done all this stuff by getting scientists to teach me in the lab over the last 20 plus years. And this piece is silk grown with fecal samples from patients in Oxford John Radcliffe Hospital who have been on antibiotics. And if the antibiotics grow blue, then they've got a lot of E. coli. And if they grow pink, they've got a lot of enterococcus. And what you'd ideally like to see in a healthy patient is a mixture of pink and blue colonies of different sizes. But you can see that some patients, so I've got a technique of growing the bacteria onto the silk and then sterilizing it, which I'll be talking about this afternoon. And so you can see some of the patients have got all blue and some have got all pink. So they've got really damaged gut microbiota. And so it's really interesting, could we repair that? And in Oxford, they created the first true antibiotic, which was penicillin. You've probably heard of Alexander Fleming, who gets all the credit for it. He kind of spotted it in the lab because he was quite an untidy person. He noticed that it was doing it, but actually abandoned the research in, I think, 1929, and it was Florian Chain in 1941 who actually got it working as a drug that they could use in humans. You think it was probably, the main story that goes out was because of the Second World War. They wanted drugs to treat the wounds of soldiers. It wasn't specifically for that. The main use in World War II was to treat gonorrhea, which is a sexually transmitted disease, because whole battalions were being laid low, of troops were being laid low by gonorrhea, it turns out. And it was very good for that. So, so this piece is the sterilized gut microbiota grown on silk. And I was interested in this phrase, make, do, and mend, from World War II, where you can patch, it says you should unpick and patch and repair things. So I patched, I cut out an antibiotic resistance gene from the whole genome of an E. coli bacteria, and I repaired it. It was an ampicillin resistance gene, which is like a modern related to penicillin antibiotic. And I repaired it with the phrase make, do, and mend, converted from ASCII code into base 4, into the DNA bases, written into the genome. And then I used that to patch a World War II women's suit that was stamped with something called the utility mark, which is CC41. It stands for Controlled Commodity 1941. And actually, it's really interesting because antibiotics haven't been properly controlled commodities. Clothing was rationed and things like that. But my dad told me that he went to the doctors in the 1950s, and the doctor said to him, would you want some of this whilst you're here? He went for something completely different. Do you want some of this? And showed him a bottle. And my dad said, I don't know, what is it? He said, it's penicillin tonic, ever so good for aftershave. So is it any wonder we're in the trouble we're in? And the little, the little sewing machine was one that my mum actually used as a child in the war. So that's part of the sculpture. And we've got the little jars there. That's the entire antibiotic arsenal, one little disc for each antibiotic that Oxford Hospital currently has. And I grew the bacteria onto cloth and used it to patch the suit. And the other jar that you can just sort of blurrily see in the distance has the CRISPR repair fragment and the molecular scissors used to do the edit. That's another close-up of it. Also, I have a project called Unruly Objects, which is all about why don't museums want to collect bioart? Are they scared of it? Are they scared it's an unruly thing that they can't cope with in their collection? I mean, I wonder if it's something to do with the term and things like that. And I worked with the University of West Attica in Athens, the Department of Antiquities and Works of Art, to understand conservation, the conservation process. And then I made this piece that is kind of maquette for it, I guess, if you like, that is carved marble with holes, with bacteria, with plants, with painting on. It's everything that's their worst nightmare. And it's also, just to be extra complicated, it's impregnated. I made it in 2020, so it's also impregnated with the extracted RNA of COVID-19 from a plasmid construct, which is a safe reagent for it, not anything infectious. And then just to make their lives easier, I've put these RFID tags in there, which contain all the conservation data for the artwork. So yes, it's complicated to collect it, but you've got everything you need to know there. One of them is being cared for in a Vinogradsky column, which is a self-sustaining bacterial ecosystem. And the other one is in the collection of the University of West Attica. And one of them sitting in Timisoara waiting to come home. We 3D scanned them as well, so there are kind of rotating 3D scans. I think Alex is going to talk about this, so I'll skip over that one. I also work a lot with other antibiotic-resistant bacteria. This is Clostridium difficile. Biofilms growing in a jar in the University of Leeds with my collaborator Jane Freeman, who I've been working with over 10 years. And what they do is they have this gut model, and they start it off with a healthy fecal sample from someone, and they let it all bubble away under different pressures and with different chemicals and things like that, different gases coming in. Then they'll give it Clostridium difficile, which is a superbug bacteria, and it's still perfectly happy, it's all fine, bubbling along, and then they'll give it antibiotics. And what happens, you get something called dysbiosis. The bacteria, the normal gut bacteria will break away from the jars and break away, the biofilms will disappear, and you'll get a full overgrowth of Clostridium difficile. And this is what it looks like in biofilms that I've grown on linen lace. I can talk a bit more about that this afternoon. But that's where the bugs live, actually. When you take antibiotics, they'll live inside biofilms in the gut. So the way to treat that is a human fecal sample. And this is the artwork that I made from it called Spindle. And I've made another work with human fecal transplants. It's actually not my gut bacteria. It's Jane and her colleagues' bacteria there. And that's on show at Eden Project in Cornwall. And we did a piece recently called Fragile Microbiome, which is made using needle felt, and I did this through participatory workshops, but all the hand beading was done by me, and the felting was made in collaboration with the scientists, so they were actually doing a bit of art as well, and we were discussing it as we made it. And it's impregnated with the DNA from the gut model, the different microbiome, and it's also impregnated with 2 bile pigments. So we were having, when you collaborate with scientists for long enough, you start having really weird conversation. We started discussing why poop is brown. And the reason it is brown is it's made of 2 bile pigments that colour it. You've got bilirubin and biliverdin. One's red, one's green. And they're also the reason that bruises are the colours they are. You know, when you get a nice sort of yellowy green bruise and then it goes brown and purple, and it's those things changing through your body. So it's like a bruise as well. So the piece is impregnated with that too. So it's talking about like the microbiome is fragile. It's like it could be bruised by the use of antibiotics and things like that. That's what it looks like. It's on show in the Musée de l'Amont in Lausanne at the moment, where they've got an amazing exhibition. Also extracted the DNA, ancient DNA from Iron Age human remains looking for ancient tuberculosis, and we did actually find it. So I made this sculpture from a bovine femur, which is anatomically prepared. And the scientist I was working with on that, who taught me the whole process for doing this, I said to him, what do you want people to know about your work? And he said, and so I'm going to tell you, he said, just let them know how bloody difficult it is to extract ancient DNA. Everyone thinks it's just like extracting normal DNA, whole genome sequencing something from bacteria, but it's not. It's bloody difficult. So there, you know, that's his important take-home message. And what I was charmed by was he said, well, we got this kind of bit of I think it was a bit of spine from the Iron Age human remains, and he cut a square from it, and we pulverized this, and we had to wash it with all different things. But he said, I'm going to cut a square. And I was like, why are we cutting a square? And he said, because I want the archaeologists of the future to know that a human did this. So that became a thing that I did. And then the rest of it's carved with the textures of how bovine TB damages the bones and things there. And then it's also impregnated with the ancient DNA. Done work with syphilis. That's my syphilis dress. Apparently, I'm the 5th person in the world to be able to culture syphilis in vitro, which is a very fascinating disease that's been with us since the 1490s. In fact, there's a rumour that is a little bit unfounded that it came back with Columbus from the New World. So that's around when it started. But it's more likely that it's probably another bacterium evolving, and it's like a little corkscrew. So when it finds an organ, it can get into any organ in the body, and it screws its way in. Cholera, I'm currently obsessed with. That's my cholera dress. Work with biomaterials as well. This is an augmented reality chandelier that I've made from working with wine waste recently. And there's a recipe book as well. I'm going into details of it. And then I'll just wind up with I'm currently working with Alex as well on a project with James Price in Brighton. I started working with him when he was a PhD student in 2010 probably, and I made my MRSA quilts with him. And now he's moved back to Brighton 14 years later after being at Imperial College and being on the National Committee advising on COVID-19 and things like that. And he's moved back to Brighton, the city I live in, because it's a very nice place. It's a bit like Barcelona, actually, in a way. I don't know, some might say. He's now heading up their infection prevention thing for the whole region. And he's looking at hospital-acquired infections. So I'm making this hospital gown that I'm embroidering at the moment with different bacteria all over it that are hospital-acquired infections, and they'll be impregnated with those bacteria. And it's a hospital gown, so you can see this beautiful hospital use only thing on it, which is quite strange. Working with Oxford University at the moment, looking at things that people throw down sinks. Because they might actually increase antibiotic resistance. It's a bit complicated, so I won't go into it, but this is my actually Starbucks coffee agar that we made, and that's that. And I was just say with James Price as well, we're currently working on a piece involving their work to predict infection in hospitals using AI. So they're creating, and they've done tests of published in the Lancet on it with COVID-19, but they want to do it for a number of different infections, and it'll sort of do a thing where it says like, Mr. Smith in bed 4 should be moved to a side room because we predict, or the AI predicts, in 4 days' time he will get an MRSA infection if he's not moved. So it's kind of like the Minority Report for infection prevention. So James thought, who can I work with that knows about AI and bacteria, and obviously came back to me. So that's nice. So yeah, 14 years later, from his PhD till now, we're still working together. So yeah, thanks. And they're my contact details, so thank you.
Thank you so much. And I will invite Josep Parelló to the... That's the defendant or the prosecution? I don't know. You prefer to sit here?
Yeah.
Much better.
Yeah.
Also with that, Okay.
Where shall I go? Where are we going? You could go in the big chair.
You can choose.
Do you want to go in the big chair?
Sorry?
You
No,
can go.
not that far. No.
No?
At least it's only
Okay.
for Jaume or for Luis, not for us.
I would do it because I'm like
My
that.
bag here.
Okay.
Can you take it, please?
Okay.
So you have
Oh, no,
space for
no,
you.
that's fine.
Good morning. So good morning, everyone. As you already know, so I think it's working, right? Can you hear me well? No? Much closer, right? Like now?
I
Okay, so
think I can use this one, can't I?
yeah,
Still?
it seems that it does not.
Yeah.
Strange. So, well, first of all, thank you, and especially the organizers, Jaume and Luis, for organizing this event, because I think that is really important to keep on keeping these spaces for a dialogue and conversation among science or arts, but I would be even more precise on artists and scientists. Okay, so in the end, so what I really like from your work, especially, is that it's always culturally embedded. So in some way, I'm just doing that. Sorry for being so direct. Is very English in a way that is keeping a lot of stories, from the past, and they are putting that on the future or even in present, at least. So these kind of conversations, that's something that I think that are very relevant. And I'm just taking some words, for instance, from Salvador Dalí, that going to the past again. So he was always insisting that he didn't want to talk with other artists. Okay, he was wanting always to talk with scientists. Okay, so at least me, for being a scientist, I also think that is sometimes, or most of the times, I would say, more interesting to have a conversation with an artist rather than with a colleague or a scientist. No, and these kind of works, I think, that are really helping to let's say think or rethink the scientific path or the scientific research efforts. A long story, but also among what is culturally embedded in the scientific effort And that was some of the ideas that, particularly in your work, is very important. No, is something that is there. No, so it's not the scientist in an ivory tower working alone and with no constant or not direct relationship with what is actually happening around him or her, or even to have something that is very much related to what the society needs at that point, which is, for instance, infectious diseases, no, which is something that is always there. No. I don't know if you want to say something on that, or what do you think about this kind of approaches, or why do you consider that space might be relevant for you?
I mean, I also in sort of the cultural relevance, because the general public know about the old stuff, but they don't know about the future stuff, or they might know about the old stuff, or sometimes the old stuff is really weird, or we know a bit about it, but we haven't really looked into it. So I was sort of touching on this thing about throwing stuff down sinks. Problem is, we have sinks all around hospitals now because we want to wash our hands. So everyone knows that we should wash our hands all the time. But most of the time, the sinks that proliferate and they aren't used for hand washing. They chuck coffee down it. They chuck Coke down it. They chuck antibiotics down it. They chuck the the stuff in the drips, the food or whatever they give to the patients. It all goes down the sink in the room, and it's not supposed to, and all the sinks are designed differently and things like that. And the reason is because of Semmelweis, who discovered this childbed fever could be like treated if we washed our hands. But what I didn't know, and none of the scientists I was talking to knew, was what happened to him. And there's this whole Semmelweis effect of people being so resistant to changing their ways, and it potentially might have driven him insane, or he might have caught syphilis, maybe when he wasn't washing his hands, from treating women. But he also lived with a prostitute and possibly had neurosyphilis, and was then taken to an asylum, beaten by the guards. This was by 3 of his colleagues who were resistant to the idea of hand washing. Took him to an asylum, and he got beaten up and died 10 days later from gangrene. So you don't hear that part of the story, and it's really interesting, this thing of this Semmelweis effect and stuff like that, which I've kind of become obsessed with in this project about sinks. I mean, when I first started, the science was like, well, what's the latest research? We're doing this study on what's down sinks. I was like, oh, okay. And then you have to sort of burrow into it and find out what's interesting, what's relevant for people, what's counter to our kind of ideas and our knowledge, because the idea that maybe we've got too many sinks in hospitals and there shouldn't be as many, or they need to be different, is like counter to our current thing. So if the scientists start saying that, people will probably be quite resistant to that idea as well.
Yeah, well, that's you were also mentioning, no, the concept of health. So well, there is many things that can be said, and starting again with what you just said, that for instance, being a scientist or even an expert in medicine on infectious diseases, most of the time you don't know the story behind all this. No, you don't know. You are not trained on receiving this kind of knowledge, or you further reflect, which is also a very important part of the story. But again, just going back to the issue of health, no, so that's something that even in medicine, no, so it's something that is now being revised, no? And I might imagine that when you're talking about infectious diseases, no, that okay, so you are not considering maybe that in a way that sometimes, unfortunately, medicine and even science, no, is considering, no, like being healthy is like is just something that everyone needs to be, and those that are just falling down are those that are that have a disease, no, instead of just considering another kind of dimensions behind that, which is much more related to well-being. About own personal stories, no, that are behind there, and that's something that at least in your work is something that for me it's really interesting, because and what is your feeling on that, and also in relation to the conversation, because I imagine that you have been talking a lot with physicians and researchers on that side, no? So if it's something that is really on the table, just to reconsider the concept of health, no? And even of healthcare, if you want. No, because you're talking about hospitals.
I mean, well, one thing is that, like with the ex voto piece, you kind of realise that no one's always healthy, you know, and that's why I'm interested in infectious diseases, because everyone's affected by them. And we, you know, what with with the COVID-19 pandemic, we all experienced how cataclysmic it can be, but also other outbreaks of the past have been so strange, so kind of important for us, like the Great Plague or the cholera outbreaks in London, which led to the sewage system being built there, which was then copied around the world. So big strides in public health, but there was so much resistance even to that as well. So, and then I've done pieces about lots of other things as well, not just infectious diseases. So I've got works around cancer, drugs that Alice is going to speak about, I think, and I work with people who, I've got this Institute of Unnecessary Research, where it's artists and scientists and philosophers, and it's about curiosity-driven, obsessive research. And we have a head of invisibility, but she's actually the head of invisible illnesses, and she has multiple invisible illnesses which she explores through her artwork. So there's that, and there's also we've got another department head. That's Bhavani Isapathy, that is head of invisibility. And then Melissa Monsoon, who is our head of microbial ecologies, but she also works a lot with fat stigma. And she's got a show on that at the moment, Sense of Stigma, because she's got a brain condition called idiopathic intracranial hypertension, which has been historically related to weight gain, but there's not really any proof of that. So it's sort of like she's just told to lose weight, but she says it's not that simple. But there's this perception that it is, and so these things are very important. So it's like sharing their work through this platform as well is very important.
Well, if you want to drive the conversation to another place, you can do that, okay? But I have many other comments.
No,
I
carry
think
on.
that maybe, and if someone else wants to raise specific questions, I'm also happy to leave the floor for that. But also another thing that the title, no, even of this cycle, is Ways of Knowing. So yes, I would think that also when we talk about art and science, it's really about that and expanding the ways how we can learn about our own realities, let's say. But I think that is also about ways of doing. Also, how in a way formats, no? How you in a way you express yourself, no? Is something that really, if we talk about art and science, and if we are talking about this sort of need of being more institutionalized or more regularly visible, no? Maybe that's something that is really one of the main difficulties, no? Because from the world of art, no, in your pieces, no, you have to follow some kind of formats, no, to be in a way, no, considered in the arts world, okay? But if you want to work in the scientific world, you have to follow other kind of rules, let's say. But unfortunately, these rules are quite different, and I have the perception, I don't know if you agree with that, that these rules are fortunately starting to be much more closer. You know? So we are not, if I'm a scientist, it's not only about publishing more papers, okay? So it's more about other kind of activities that are just more in this cultural kind of sphere in which culture or arts has already a place. No? So I don't know how you feel comfortable and uncomfortable, no? With these kind of codes, no? That we all have, no? And which are very distant, but at the same time, of course, you have to work, and you have to work as an artist, even if you have a residence maybe in the end, no? And you already mentioned, I have to do, or I will do an installation, I will do a piece, no? I will produce something, no? So that's why I think that also ways of doing is something that we need still to think better about, and how comfortable you feel in that situation, and where are the conflicts and where are the challenges.
I mean, the first thing I would say is it's this idea of being embedded in the lab and doing the work alongside the scientists, and having them teach me and me doing it, because I don't think that I can explain it unless I've done all that and I really have a good understanding of the process. So there's that. the thing I say with the Institute of Unnecessary Research is that art is a meta-discipline, and it can bring together other disciplines. So you can bring science, and it can bring history, and it can bring philosophy, it can bring ethics, all together in the art object, if you like, if it does result in an object. I guess the artist is the one that mediates that what that object will be. But I mean, a lot of the scientists that I've worked with have helped facilitate the exhibitions as well. So sometimes the galleries are interested in working with the scientists, and then they say, well, we're working with the artist, and then that happens like that. So my big show at the Museum of History of Science in Oxford came from my science collaborator talking to them, because she was so enthusiastic. So there's that. And then I think I'll tell you a story of, so when I had a solo show back in 2014, and I had a budget for a PR lady. So this was great for so somebody who can do all the press and get me on the TV and stuff like that. And I got on BBC Global World News TV, a live interview through her. So I had all the professors from Oxford turn up to the show. And the Guardian newspaper came, and they interviewed my longest collaborator, Professor John Paul, that I'm still working with. And they asked him about this, and he said, well, you see, the thing is, nowadays you have to do public engagement in science. And I looked at him and was like, don't say that. Why are you saying that? And he said, no, but what I mean is, it's the perfect excuse. So nowadays they want you to engage, like the scientists to engage the public. And that means that you can work with artists, and you have great fun and explore these stories and things that you wouldn't necessarily directly explore through the work, and have exhibitions that get on BBC Global World News. So I had the professor of microbiology from Oxford University turn up, and he was like, well, if this is doing better than our papers are doing, this is getting us more publicity. So it's getting them out there as well in a bigger scale. So they love that. It's quite funny.
Yeah, no, that's the kind of conversations that are starting to, maybe not very in a sort of utilitarian kind of way, but that's something that is already in the air. That's my perception right now. No? And also considering that kind of approaches. No? So let me just be polemic. No? So let's say, okay, so we have artists in our labs, but when we can have a scientist in our galleries? So
but they do have that.
They already have.
Yeah.
Do you know some examples that we can share here? Or
I think the Gluon project in Brussels has facilitated that. They've definitely had scientists in labs. So it was an EU-funded thing. I think it was related to the STARTS program of the EU, but it was, Gluon project did it.
Well, it's still marginal. In any case, I know another case. I don't know if you know. There is a scientist that is called Barabasi that is working on complex networks.
I know him.
And he's doing some stuff now with galleries, almost.
Oh, yeah, no, I've seen it. It's lovely,
not to publish more papers, almost. I'm just exaggerating right now, but it's more in the business, let's say.
He used to do a conference called Complex Networks in Art and Science, and I presented at that a few years back in his lab, so...
But I'm talking about a very kind of perception.
They're beautiful as well. Also, he's done this amazing book that works out what artists are going to be successful based on the first galleries that they showed in. But he won't share his data set, because I did ask. I wanted to see where I came in. I'm sure I'm not very good, actually.
Yeah. So another thing, just going back to the initial discussion. So we met some time ago. And you were mentioning the case of Picasso. It was 10 years ago. Okay, so it's a long way, no? And you were talking about one canvas that unfortunately is not so much underlined in the case of Picasso that you said, no? That there was the person laying in bed. And I think that if I'm not totally wrong, so there is the science represented by a physician, no? And also how the art is trying to represent that. And this was the starting point at that point to invite you to come to Barcelona and try to work on that. No? So Where I'm going, I want to go with that. So I think that now maybe again, we're talking about Picasso, we are talking about other sort of artists at that time that they were truly fascinated with science or scientific progress, or many of the conflicts that were happening now at that time. That it seems to me also that now there is again this need not to start, to represent the scientific world, to express the scientific world, not to put the scientific world on stage, because that's something that in our society is also, even to criticize and to put, to put there and to shade some lights, as you said. So that's something that in a way is motivating your work, or just the curiosity, as you just said, how you see science as a cultural phenomenon, let's say.
I mean, I think sort of from a political with a small p kind of perspective, I think that the wider public need to be able to engage with science, and the way they get it now, I mean, when we were doing that, the way they got it was the newspapers. Even we're talking 10, 20 years ago, it was the newspapers, and they want to sell papers and they want to keep you going back. So they would give you only, like, if you read a newspaper, it's the last paragraph that has the actual facts in it. Not, you just skip, go headline, last paragraph, and you might as well, because it's like shock news, like dirty hospitals in the UK give people multiple superbugs. Actually, it's a different strain circulating. It's new. It's been whole genome sequenced. It's a completely different story, but they'll say one thing. And now we have the social media phenomenon, which obviously, and you've got so much different false stories. So people don't even know where to go or who to ask, because who are you going to ask? You put it on Reddit and you get like 50 conflicting answers or something like that. So having these art exhibitions where we do events with scientists as well, and people can kind of come, like I really like participatory workshops as well, and I do them for all ages. So I don't do kids workshops per se, if I can help it. I do them for all ages. So children can come, their grandparents can come, they can tell stories about, I had one where I was doing a workshop in the Victoria and Albert Museum in London, and there was a lady there who'd actually been prescribed as a child a sulfonamide drug, which is a precursor to an antibiotic, and she told that story, and we incorporated that in ex voto and things like that. And then little children who, you know, have done these workshops and come back over and over again and tell their parents. I asked this little girl, she was like 8, came to my workshop multiple times, and I said, well, what are you going to do when you grow up then? I was like, when she'd say artist, but she said synthetic biologist. So, you know, that's and the parents engage because kids engage and things like that. So then people have a way in and they can critique things and they can ask things. So when I was doing the workshop at the V&A, I had James Price, I had Professor John Paul, all sitting down making MRSA quilt squares together with the participants. And then when you're just doing something, a bit of sewing and putting something in a petri dish, you can just chat about things because it's not a high pressure thing. So they're asking, the public are asking top scientists these questions. And would you go to a science festival, great big audience, stand up the back, say, excuse me, how does that work again? I didn't quite get it. But you can, in a workshop, you can ask that, and you can, so it's about widening it out to people and having different methods. So there's the talks, there's the gallery tours, there's the hands-on workshops, there's the videos of the gallery tours and the talks and things, and all the writing as well. And sometimes I write scientific papers as well. Just written one, actually, just published in Applied Phycology, of all places, the top seaweed journal. Watch out for that one. So it's actually open access, so as well. Didn't get paid for it because I'm an artist, but they did waive the £700 open access fee for publishing it, so it can be open access. So
that's another discussion from the scientific world. It's quite conflictive as well.
Giuseppe, sorry, if
we was going to close.
Yeah, we have a couple of minutes left, so perhaps let's consider. I mean, I would say let's do just one question, but I give it to you. Perhaps we can just give the chance that the last comment question is on the audience, if someone is willing to.
I think that
yes. Jaume, please.
Thank you very much. Fantastic. Anna, my question is very simple. We have understood your thoughts, your intentions, the means of explaining and what you want to explain. But at the very end, you end up with an object. How you get the object and to which extent aesthetics is important for that.
Aesthetics is like totally it, but not just beauty, but also disgust, horror, fear, the sublime, aesthetic concepts. That's at the basis of everything I think I do, and I think at the basis of everything that scientists do as well. So is literally kidnapped by my collaborator Jane Freeman at Leeds University for 4 hours, where I was explained how much they love Clostridium difficile bacteria. They love it. I mean, you don't think about loving a highly pathogenic, deadly bacterium, but they love it. And so these aesthetic sensations are always there. That's why I use the real materials in the work as well. I think there's a sensation that you get from knowing that that's actually Yersinia pestis DNA on it and that I extracted it in the lab and I'm an artist and I was allowed in to do that under extreme circumstances. They don't just let any artist in, but I've been working in this way for so many years. I've built up these contacts and I'm now able to do that. So that's very important. We did actually publish in FEM's microbiology letter, Federation of European Microbiology Societies, with Nicola Fawcett, a paper about this called Bacteria on Display, Can We, Should We? So that's out there. So we do have these, like, usually quite short papers, I admit. But,
Okay, thank you. I would. I'm really curious to hear Marta's question, but perhaps we can keep that for later because we need to move on. Also, we need to let Josep go because you have your class in a few minutes.
Well, if you want to come to quantum physics class, I think it's much better to listen to Marta's work. Thank you very much.
Thank you very much. Anna Dimitriou, Josep Parollo. Thank you. Now I will invite to the stage Marta de Menezes. And she will be in conversation with Lua Koderk, and then we have the coffee break. You probably know Marta. Marta has been active in bioart since the 90s. It's probably fair to say that she's a pioneer in the coming together of art and biology. Marta is the director of Cultivamos Cultura. It's a quite active organization in Portugal devoted to experimental art. Check that if you don't know it. And she's also involved in Ectopia. That's a project meant to facilitate the collaborative work between artists and scientists. And she will be later in conversation with Lua Koderk. Lua Koderk is an artist. And she's here today because she's been involved for some time now in a project together with Mafe Moscoso and together with Mariona Moncunill in a project in Bau. They are collaborating with ICM, the Institut de Ciències del Mar. And it's a strong project that we will hear about collaborating artists and scientists. So thank you, Marta. When you are ready, please go on.
Okay, is this working? Yes, it is, right? And thank you very much, Luis and Jom, for inviting me to participate here. It's a pleasure, and thank you all for coming. It's always good to see people in the audience, and I hope we have a nice conversation. So I don't know, maybe I should start by saying as well that I'm trained as an artist. I never trained in a professional capacity as a scientist, but as my scientist collaborators like to say, I'm a very proficient technician in the lab. And if you're a scientist, you're most likely to understand what I'm saying. If you're an artist, maybe this sort of goes a little bit over your head, but I hope we will have a chance to discuss this. I'm going to talk about two projects that I did relatively recent. They're not my latest projects, but they're important to me. But I would also like to introduce you the two institutions that I artistically direct in Portugal. One is Cultivamos Cultura. It's in the south of Portugal, 2 hours south of Lisbon. It's in a rural area and in a natural park. And it has a very robust program. We have about 20 residencies per year of people from all fields, not just artists. And even within the arts, it's not restricted to artists working with science at all. But because we're in a natural park, people tend to work mostly with ecology and sustainability and anything that has to do with the specific climate and characteristics of the area, the ecosystem, and the species that are involved in the environment around. Cultivamos Cultura, it's an old farm, even though it's in the middle of a village. And besides the residencies, we have, of course, exhibitions, both nationally and internationally. We have conferences and workshops, and we also teach. And so it's quite a robust program yearly. And we also have something that is not very common, which is a collection. Let me just make sure that there's no sound coming off of this. Yes, it is. Well, at least it's in the computer, so it's not going to completely overwhelm you. So one of the things that we have is a collection of art that is difficult to host, to keep, to store, to archive. And so we started, we're not a museum, we don't have a permanent exhibition space to show our collection, but we wanted to make the collection available to the public. So we have actually applied and been given a few years ago a European project to create this platform online with 4 other institutions in Europe, and created this online platform called Archive. And one of them was actually here. It was Hangar in Barcelona. And Luis was leading it at the time. And there we have made available a lot of projects in art and biology, including our collection, and in form of documentation. So it's not a digital exhibition space or anything like that. It's meant to be a challenge in terms of documentation of these artworks and about the history of these artworks, how they've been exhibited in many different places around the world. And for our specific collection, because we have the residents come and give us an art piece, we have the ultimate opportunity to give the voices of the artists to the documentation of the piece itself. So we always make a small documentary of the residency where the artist is given a chance to talk about the process and the reasons why they're doing this artwork in the specific space. And it has been, for us, it was a discovery that the potential of having these small documentation pieces next to the exhibition of the pieces has been really amazing in terms of engagement of the audience with the works. So it is one of my favorite modes of documenting these works now is actually these small video documentaries. The other institution that I run is in Lisbon. It's more focused on facilitating interactions between artists and scientists in labs. Of course, in a rural space, you don't have that many laboratories, except field laboratories, which is great. And most of the biomedical and biological, and well, most of the labs, scientific labs, are based in big cities. So Lisbon is one of them. And after I came back from Oxford and then Australia, where I did a residency at Symbiotica, I wanted to also help facilitate interactions between artists and scientists. And we decided to create Ectopia. Ectopia has an exhibition space, which is what you see there. What you see there at the top is the exhibition area of Ectopia. At the top or at the bottom of the slide, you see a little bit of the lounge area upstairs and library that we have. It's also my office space, and also where artists stay when they're working with scientists in their labs. So it's a little bit of a small apartment where the artists can stay and go work in the lab with the scientists. We have a close partnership with the Faculty of Medicine and the Molecular Medicine Institute, which is associated to the Faculty of Medicine in Lisbon. And with them, we have ongoing exhibitions in the institute, in this little pod. So about every 8 weeks, we change the artworks that are in exhibition there. It's in the main lobby, so scientists and students, it's the Faculty of Medicine. The students are always moving around, so everybody gets to see these artworks and enjoy them. And it was part of the strategy to make sure, because they do have quite a lot of artists in residency at the Institute. It's growing more and more. And so it felt to us as a collaboration that it should be something that everybody should have access to, and not just the scientists in the labs. So our strategy was, how do we show what's being done, and how do we make it a regular presence in everybody's lives? So we have exhibitions, we have roundtables, workshops, and of course the residencies, and everything is on the website, which is what you see here. Now, just to give you an introduction about me, like I said, I'm trained as an artist, but actually my final project for my painting degree, which was a very classic painting degree from the Faculty of Fine Arts or the School of Fine Arts in Lisbon in 1998, 99, I don't remember anymore, was the project with the butterflies, which most people actually know me for. And I was very thankful to my studio painting professors to actually allow me to present this project that I did in a science laboratory as my final project. They graded me not very well, and I'm not sure how they feel about that, seeing that it was so well received in the experimental art field at the time. But it's always a nice story to tell, and I am thankful that they didn't make me paint butterflies instead. Because probably they could have for me to finish my degree. But since then, I've been working in laboratories in many places around the world. For about 25 years, I would say. Yes, actually, I would say no, I'm absolutely positive. For 25 years, this is 2024. And actually, it's the 25th anniversary this September that I presented this as my final project. So I've graduated 25 years ago. And along these 25 years, I've had the privilege of working with many different scientists and making friends with them, not just working as collaborative partners. These are some of my best friends. They were engaged in my work, and I always felt very lucky that they were so engaged. A little bit like Anna, it's really a privilege to understand how excited they are about the work that my crazy mind comes up with in terms of proposing, unthinkable things, and it allowed me over these 25 years to understand also how I work and what really interests me. So over these 25 years, I've discovered that I am really fascinated with one particular subject. And I used to say to my colleagues and friends who were scientists that I would be a horrible scientist, and I still think that. Mostly because I couldn't focus on a specific subject. And if you want to be a scientist, you really need to be for a very long time, basically your whole career, to be completely focused on a specific subject. And I never thought that I could until I realized that I actually am. And that subject is identity. Okay, so for some reason, I've come to realize that I am absolutely fascinated with the concept of identity. And I find that biological sciences, and especially biomedical sciences, over the last probably around 100 years, have generated knowledge that really challenges preconceptions of what we have understood as identity. And for me, this is absolutely fascinating. This is why I work with biological sciences mostly. It's because they keep challenging what I understand as identity. And yes, at the beginning it was probably very naive, and it was very, I guess I have been through the idea that identity is a core, that it's a fixed thing, that I am who I am. And nowadays, I will tell you, I'm always changing. There's no such thing as a fixed identity. I am transformed every time I'm in house or I'm outside of the house. I'm transformed with every conversation that I have. And I guess part of the work has evolved to evidence really this understanding, this re-problematizing of what are we and who are we and where do we come from? And more importantly, and this is why I think it's so relevant, understanding all of these steps allows me to try and think how to choose my future, who I want to become. And I do think that this is very important, because not only at a personal level. So yes, I want to know who I am, and I've probably always wanted to know who I am. And I've probably always felt a little bit outside of the box, whatever box you can think of. And I always thought of myself as someone who thinks weirdly and not exactly the same way as everybody around me, which I've come to realize that it's a box in itself. So there's lots of people who don't think like others do, which demands the question, does anybody think like others do? But again, this is why I feel it's so interesting. And so it can be a very personal thing, but I do think that nowadays in a society and in a world where individuality is so relevant and where community is so relevant as well, we need to understand identity as a complex system that plays with the individual, but also is very much rooted in this idea that we are more than just one, that we can never be just one. And this is why I think identity is so important. And this is why I roam around very different fields in biology, because they all show me an aspect and put a question that is different. Those are the questions that I'm trying to pursue. So I have worked with microbiology. I have worked with evolutionary biology. I've worked with physics and medical imaging. I have worked with software. And many different fields. I've also just done things like growing plants on a specific page of the Bible, and very, very not cutting edge science at all. But more recently, there are two fields in biology that I feel really challenge profoundly my understanding of identity. One of them is, of course, genetic modification, and this understanding that it does change actually mostly came when CRISPR became a thing. So before this, I did understand genetic determinism, and I still think we're very much attached to this idea that genetics is a key. And that's one of the things that I like to challenge most of the time in my works, is this: why do we keep understanding genetics as a book or as a set of rules when if we dig a little bit deeper, and the knowledge that we have nowadays actually challenges this all the time. When CRISPR came about, it sort of put questions to me that I could revisit the Butterfly Project all over again. You know, what is considered natural and what is not, and how can we determine traits of organisms with this revolutionary technique, not even a conceptual revolution, but a technical revolution, which was really interesting for me. But of course, a subject in biology that is very close to my heart, and you will understand why soon, is immunology. And I never thought that immunology would be so challenging in terms of identity, mostly because I didn't know very well what was immunology before I met my husband, and we did meet a long time ago, and we got married 25 years ago as well. So I have had a very long relationship with immunology indirectly, and only started working with Luis in art projects from 2014. So I worked with almost any other field until I found a project that I could, or that made sense to work with Luis. And I suppose still hearing about immunology and hearing about his work kept giving me questions, but the level of complexity is so high that I never felt very comfortable in actually pursuing that. And if there's anything about immunology that most people understand, is that it's extremely complex. Immunology conferences are the ones that no other scientist goes to because they know that they won't understand what they're talking about. And it's a joke in science as well, because yes, they are, for me, it was actually a revelation, and it's kind of a refreshing attitude. I don't know any immunologist who will try to make things simple for you. And working with scientists for such a long time, most of the time I have to overcome the desire that they have to be understood, and which gives them this drive to make things simpler for us to understand. This is something that I usually have to overcome. It's like, yes, thank you, I understand the basics, I did my homework, so please give me the complex, because I know this is not that simple, and this is actually what I love. I have never had that problem with any immunologist, because they know that it's not simplifiable. There's no such thing in immunology, and we actually know so little about it still, that it's not possible to generalize things or to have a rule or anything like that. It's basically a complex system of interactions, which is for me really fascinating, and also gives me so much room to try and use as metaphors, as analogies, as modes of trying to understand the complexity of what identity is. Because one of the main concepts of immunology is the idea, or the understanding of what they call self and non-self. And how can this not be about identity itself? So the first work that I did in immunology with my life partner was Immortality for Two. Immortality for Two is not an immunology technology at all. It's about cancer, which in some ways is about immunology, but it's about how we named what is effectively cancer. When they discovered these groups of cells that would divide indefinitely, that wouldn't stop dividing, they decided to call them, or scientists decided to call them, immortal cell lines. And actually what they are is cancer. So by definition, a cancer is a group of cells that won't stop dividing. I found it really fascinating that they would name this anything close to immortality, because a very mortal affliction is related to our idea of identity, of immortality. So this was absolutely fascinating for me, and I decided to generate a process of trying to understand this apparent paradox or apparent disparity of understandings by proposing to take my own cells and make them into immortal cell lines, which by 2014 was possible through simple techniques, viral vectors and inserting oncogenes into potentially any cell. But of course, it's for health and safety reasons, it's not allowed to. So I cannot work on my own cells and actively transform them into a cancer because there is a possibility that I may reabsorb these cells and my immune system will not differentiate them from me, and I would effectively gain cancer. I'm very glad for those kinds of rules. I promise. But also, I want to make sure that people understand it's not an ethical issue in this case, it's a health and safety issue. And a lot of people don't really understand that this is very different. It's not because they didn't want me to do it, it's because there is a health issue that is raised if I want to do this, and scientists are not allowed to do this for this specific reason. So I was stuck, and the project evolved into, why do I want to understand immortality in this specific way personally? Why do I want to achieve any kind of immortality, whatever that means, through creating a cancer of my own by myself? Who wants to live immortal, whatever that form may be, by themselves? And in our imagination, actually, if you think about it, what are the immortal beings that we have imagined as humans? I think the first ones, well, I don't want to offend anybody, but the first ones are gods. Gods are immortal, right? And we did imagine them. Right? I don't think anybody has witnessed them. So whether you are a believer or not, it was some way. It's our concept of immortality is represented into these beings. And of course, not just gods, but superheroes, that some of them are immortal. So for gods, they were never human, so immortality comes attached with the absence of humanity in a way. For superheroes as well, so they lose their humanity in a way. They're no longer just humans, there's something else, something other, a little bit monstrous, of course. And then you have the monsters that we affirmatively denominate as monsters, like the vampires and the zombies and all of those creatures that we created in our imaginations that have lost mortality. And so mortality is very much associated with the identity of what is human. And this, for me, was really a fundamental part of this process. So I proposed to do this with my life partner, who, would you become immortal with me? And we said yes. And so this is also one of the reasons why we decided to use our cells from the immune system to make ourselves immortal. And the health and safety problem didn't exist anymore, because I could transform his cells into immortal cell lines, and he could transform my cells into immortal cell lines. And this is what we did in Luis's lab. And in this project, and I suppose we're here to talk about collaborations between artists and scientists, and this is also why I wanted to speak about these cases specifically, because I'm not just working with any scientist, I'm working with my life partner. So unavoidably, the projects became very personal as well. And because of this desire to, okay, I want to share immortality with you, I actually love you, so I wouldn't mind spending not just the rest of my life with you, but the rest of existence together. So we did this, and in this particular collaboration, I was very much involved in my job in the collaboration, and in the negotiated collaboration, was how to make it into an art piece that can be shown in a gallery space. And for Luis, it was, okay, what are the protocols and the techniques that we need to work in the lab to create these two cell lines and to maintain them? So how do we store them? How do we revive them? How do we propagate them for exhibition? It was very clear that his job was one and my job was another. It was a very successful collaboration, and we do love the work very much, and it's very meaningful for us to have this ironic piece where, despite wanting to share immortality together, because these are cells from the immune system, we can never be in the same place together. This is the immune system. They will kill each other because this is what they're meant to do, to differentiate self from non-self, and there's no tolerance to the non-self in this case. So the piece is shown with the 2 cell flasks with the live cells in opposite sides of the table, and the only place where the 2 cell lines can be together is digitally on the overlap of the 2 projections on the top of the table. And this is to stress out all of this irony of sharing immortality and creating these 2 lines of cancer that are our way of becoming immortal. And how it's still impossible to be together, and so there is a price to pay. More recently, we've developed a project together that I absolutely love as well. As you will probably understand, I love most of my projects, and some more than others, and these are some that I really engage with, also because they're incredibly personal, that actually changed our way of collaborating, and I'm very happy about this. And it was something that I observed towards the end of the project. So together we went to see this presentation in Lisbon by John van Rood. He was the father of clinical transplantation. He was a very important scientist. He was a medical doctor. Besides being an immunologist, he was one of Louise's heroes. Louise actually did his PhD at the then School of Pathology in Oxford, where Florey did most of his experiments, and where they treated the first patient with penicillin, who died a few days after the treatment, which they don't usually tell you, as Anna would say. A few weeks. So John van Rood, who was a Dutch scientist, was one of Louise's heroes because he was a very important transplantation scientist. And the reason why we have clinical transplants today in hospitals was because of some of the research that he did in the 60s. And we went to see a talk that he did in Lisbon, actually the year that he passed away. But he was an amazing speaker. You know, at this time of his life, he couldn't care less if funding was coming, because that was no longer his concern. And so he could be a lot more open and a lot more funny about the things that he did in the lab, especially in the old days of his research. And the experiments that we are actually talking about or referring to were experiments that were done in the 60s in Holland, where he convinced all of his colleagues in the lab, which would not be possible nowadays, to have small skin transplants and to follow the transplant rejection. And this is actually what gave rise to understanding HLAs, which are human leukocyte antigens. And this is what gave rise to a proper matching between donors and recipients of transplants. He was also one of the founders of Eurotransplant, and this is a picture that actually is in the paper that they published, where you see the colleagues looking at the rejection. So the transplants were, this is a self-transplant, so there would be no rejection for this, and then you would have 4 transplants from 4 colleagues that would expectedly be rejected, and they would follow up on what was happening at a blood level. So what are the levels of antibodies and all of the reaction that the body produces from receiving a transplant that is not their own. So this was absolutely fantastic. There's a lot more stories around this, including asking the Vatican for access to records of cousins that got married because they needed people who were genetically closer to do these experiments on to see if there was anything that was different. when he was talking about this, and after this presentation, Luis and I came home, and we thought he will say that I came up with the idea. I keep in my memory it was him, so I don't know, because we know how memory works. It's not exactly factual most of the time. But we came up with this idea that we should do a transplant between each other, and we should reject each other. Now, if you know me and Luis, you know that we always have crazy ideas. You know, this is why we are together and have been together for so long. Luis is in no way less creative than I am, or less wild in the ideas that he proposes to do as artworks, which was always something that he did, which always fascinated me. So he always comes up with these crazy artworks, and most of the time he wants me to perform them. And I keep usually telling him, sorry, no, that's not a Marta work, that is a Luis work. You should do your own artistic work. But in this case, it was the exception, because this idea actually felt to me that it was also my idea. It made sense in my body of work. It made sense in my mind to use this as a way to understand a little bit better what is this concept of self and non-self, and what am I doing by rejecting my life partner biologically. You know, it's not that I am rejecting him, I'm not divorcing him, I'm not doing anything like that. And I don't want to, but I know that there's no way my body would accept having a part of his body inside of me. Or in this case, on me. And so we did do the surgery, and we did as expected, reject each other. And I think this is where the collaboration shifted, or the nature of the collaboration shifted, was because we had such different views of what was important in terms of concept of this piece, that it made me realize that he was no longer in charge of only the scientific part of this. Actually, he was not in charge of the scientific part of it at all. It was the surgeon who performed the surgery. There was no scientific research involved in this. For him, it was such a different project than it was for me, that it was really beautiful to see how this was the moment that I said, yeah, you're the author here, and I am the author here. And our works are almost different works, even though they're formalized in the same piece. And for me, this is beautiful. It was sort of an achievement for me to actually engage so much in a collaboration with a scientist who's not trained as an artist, that he's creating a work, and it's a really relevant and very good artistic work for me. So let me explain what this means, okay? I am going to not turn on the video just yet. So for me as an artist, my concept is identity. So this idea of self and non-self was the focus of the project for me. This idea that I was physically rejecting a person that I choose to live with is what is challenging for me. That my body knows who I am despite my brain. So for me, identity until then was very much understood as a consciousness thing. It was a cognitive thing. And what this work allowed me to think was, no, it's not. Your body knows who you are long before you are aware of yourself. And this is true. The immune system is there before you are born, and it knows to differentiate you from anything other before you are born. How does that challenge your identity or your idea of identity? It doesn't really challenge your identity. It challenges your idea or understanding of identity, is part of what is interesting for me. In this specific project, as someone who has lived for a very long time with someone and who is a contrary person, and I can guarantee that I am a contrary person, I'm not an easy person to live with, I need to affirm my identity on a regular basis. And Luis is the poor guy who has to deal with it. So I rebel every now and again. And I say, I am not us. I am tired of being us. I am me and you are you and we are together. Yes, but I am me. And it's sort of, this was my moment to say this with my body and not with my voice or with my actions. It was, my body is telling you and affirming myself through the rejection of this person that I choose to live with for the rest of my life. And this is the core of the piece for me. And this is the main concept that is challenged. This is the question that I want to put forward is, who are you when it's not just your brain that tells you who you are, when it's your body that knows who you are before you know yourself? For Luis, because he's an immunologist, the concept is completely different. And fortunately, he's a much more romantic person than I am. And so for him, the rejection is a temporary thing. And you know, that's probably why we're still together, because again, my rejection or my self-affirmation is usually a temporary thing. For him, the rejection was a temporary thing. It was actually the most, not inconsequential, because the consequences are the fulcrum of the piece for Luis. As an immunologist, the rejection of the skin, it takes a month and then it's gone. There's nothing more there except the transformation of the body. The body creates things in itself. It becomes something else. It changes who it is because of the effect of this rejection. So as we are rejecting each other, antibodies are being made by our body. And the cells that are producing these antibodies have been genetically manipulated by our immune system to have DNA that doesn't exist in any other cell in our body. And so these cells will have a different DNA because I rejected Luis. And I have these cells inside me for the rest of my life. And so it's more than a blood pact. It's in a way, it's this, the self-affirmation has a very important consequence, which is a transformation in myself. This is why I say that I am always transformed, that my identity is always different. And in this particular piece, that identity is so forcefully transformed that in my body there are cells that have DNA constructed by my immune system that are a specific consequence of this rejection, of this reaction, of this affirmation of self. And for Luis, this is the most important part. This understanding that the immune system is not just, it's not about throwing away a thing that you don't think is you. It's about how that thing transforms you, how it becomes a part of you. And for me, this is the most beautiful, I fall in love with the piece, the way he understands it. I'm not going to show the video. If you want me to show the video, I can show it later. I don't want to have more time taken. But this, just to let you know that this then started a series of works that I am going to do with Luis in the future. One is distributed me. So again, how many types of cells can I create with an original cell from my immune system? So if I take one of my B cells that was created by my immune system, has the DNA that was manipulated by my immune system, and undifferentiated, how many types of cells can I create from that? Can I create another body? And how does the identity of this other body, which is a disembodied body, how does the identity of this other body relate to my own identity would be the main, my main question for this project in specific. And of course, after achieving immortality and then rejection, I think the obvious next project with Luis would be tolerance. How do you achieve tolerance? What does tolerance mean? Whether you think of it as an immunology concept or as a social concept. How do we understand tolerance? What is tolerance for us as humans? So just to finish off, I want to thank all the scientists I've been working throughout my career. And I always remember them fondly. Some of them still have me on their website as their artist in residency, even though I've left for many, many years. And thank you for listening.
Thank you so much, Marta. Now I will invite Lua to the, how you want to do it. Perhaps you can do as
Sure.
Anna and
We
Josep.
can do the same.
So that's more like a conversation.
Not step on any computers. Now I realize that this is actually double. You have a pillow on top of the seat. Really comfy and high. Okay. I know. One more. Nobody wants to. Nobody wants to sit there.
Okay.
Very well.
Okay.
Thank you.
Well, first of all, thanks for having me, and thank you, Marta, for your presentation, which was really inspiring.
Thank you.
I wanted to talk with you about. This is some idea I've been like thinking of for some time now. I want to talk with you about two dimensions that always coexist in any art science project. And this is something that I connect with something that Stephen Wright calls a double ontology.
Okay.
Although I think also that those two dimensions are found in different proportions in each particular practice. And each dimension leads to different ways of knowing also. The first dimension has to do or deals with the issue of representation.
Okay.
And which, you know, it's a main concern in the art tradition, and has to do with the metaphorical power of what you do, and also its symbolic power and narrative issues as well. Yeah. The second dimension has to do with how art practices adopt the ways of scientific practice. So it has to do with how art practice operates as science somehow.
Okay.
And again, thinking about what Stephen Wright says, that means that your practice in a way would operate on a one-to-one scale. So it's not only, I mean, it actually is something that affects the world. No, it has well. So the thing is that I think your practice, for me at least, has a very powerful equilibrium between those two dimensions.
Okay.
And for instance, I was thinking about that piece about the transplant. I don't know if that makes sense. For you, to you. But of course, you're talking about love, identity, and the thing you were talking about on the last part of your presentation, which is something that really moved me, is about that narrative and that symbolic part of that piece, and how it was different for you and for your husband, no? The part that you were stressing and the part that he was like putting value on. And I would like to know, because I also think that connects with what Jaume was asking about, the aesthetic value of your work and where it places itself in the world. And that has to do with this idea of a double ontology, because this is something that makes sense in the art world, but also makes sense in the scientific world as well. Its value, it's not only scientific, artistic, sorry.
Okay, I'm not sure I'm able to answer very much that question. It's not really a question, it's a provocation in a way, right?
Yeah, kind of.
Which is good. I like that. So again, I remember I said that I've been realizing over the many years of my life that I think differently from most people. I'm still very much convinced of that, because a lot of the time when I read, especially when people write down things that if it's not science-based or fact-based, if it's a matter of reasoning, this is when I realize that I don't think in the same way most of the time as many of the people that I read. So I'm having trouble even trying to understand exactly what you were referring to as the two dimensions. I'm not sure I understood what they are. So yes, of course, there's I can say a lot of things of what I understand in terms of art and representation and how it is a very complicated subject. And of course, art and biology or bioart, if it has posed or challenged that conception greatly, because you're not just representing life as you would traditionally, but you're using life itself. And the way I say it, or the way I try to reason it, is actually bioart is using life to represent itself. It's not that it stopped being a representation. It's using the material itself, the life material, whether it's alive or not, to represent itself. And this is, I think, also one of the things that Anna was saying. There's something really moving and challenging to your understanding of what something is if you know that it's the real thing that's there, whether it's active or not, whether it's alive or not. There is something different. And this is, I think, the major challenge that art and biology pose to this idea of representation in a classical sense, through this dimension of art being about representation. What happens to the idea of representation when you're using the thing that you're representing to represent itself, right? When you're making that circular movement. And it's, I think, in a way, a very ironic way of doing it, which, if any field of knowledge would do it, it would be art, right? Because that's what art does in its nature itself. It's to challenge whatever preconceptions of what you may think art is about. But also, it's not really challenging. I don't see it as really challenging that much, because it's still about representation. It's in this case, I don't necessarily think that art may be representing life. It's representing what we understand as life. We've always been wrong in thinking that it's representing life. No, it's representing an idea of that we have of what is life. And so using life to challenge what is the representation of life, for me, makes absolute sense. It fits. This is where it should go, because this is who we are. We want to challenge. Now, this is how knowledge evolves in a way. It's, if you can give an extra dimension to what has been accepted already, then this is when, at least in the arts, from my perspective, this is adding knowledge to the field. This is the core to what creating knowledge to a field is, right? This is the definition of creating knowledge, is that you add something, and by using living material and life itself to represent our understanding of life, our knowledge of life, then that is adding something to the field. So for me, in terms of knowledge, now I didn't understand so well the second dimension. Maybe you can...
Yeah, in the sense that your art practice uses the ways of scientific practice. I mean, you can use science only thematically as a theme for your work?
Really?
Just yes, it is possible. For instance, you
Of
can
course.
make a painting about microbiology, yeah, about, for instance, that, or about a microbiological life, or, but you're not just doing that. You're kind of adopting the way science operates to do your work.
No?
And for me, this is something, I mean, this is something that is happening since the 60s. The way artists found the necessity to get out of the art bubble, no? Operating outside the autonomy of art and kind of relinking to life, no? And kind of finding ways to relate to the world differently. And I think your practice is doing that.
Okay. I don't know. For a moment there, I felt more like this is a challenge to what we understand as science. And again, that I think relates to what Luis was saying at the beginning, that nowadays it feels to me sometimes, and it's not how I understand it at all, which again reinforces the idea in me that I don't think as other people do. So for me, I never acknowledge, because now I know people do think that, and I feel very angry about that. But I never would acknowledge that science is the sole proprietor of knowledge.
No.
So it just doesn't make, that's not how I was taught. Philosophy might, at some point in history, have been the one of the major holders of knowledge production, but it was never the only one. And since science came from philosophy, which I usually tell scientists who think that philosophy and science have nothing to do with each other. Well, actually, if you go back in history, science was not even a thing in the Renaissance. It didn't exist as a concept. So knowledge is much larger than any of the fields that contribute to it, right? And every field, whether it is fact-based knowledge as science, not just, I can't say just it is, but this is the core of the scientific method, is to try to be based on facts, to build their knowledge on facts. And this is part of the identity of scientific knowledge, which again reinforces that there's not only scientific knowledge, there's many other fields of knowledge that are not science-based, including the arts. I don't see where in history did we make the mistake thinking that the humanities was separated from the arts. Arts is a part of humanity, and so that the arts would not contribute to the field of knowledge. I think this was a mistake, and we know better now, so we should stop thinking that way. I usually think, and maybe this is something that we can discuss later, I usually think that the different fields of knowledge are in different positions, or in their value in society is usually in different rankings, which is something that I'm not very happy about, obviously. But if you think of knowledge as a whole, scientific knowledge, which has it in its very nature, the fact searching, this is what I usually say that is very different between the arts and the sciences, is that the arts is the only field of knowledge where the individuality of knowledge is absolute and valuable. It's where consensus does not exist or should exist. Your interpretation of a piece, and therefore the knowledge that you produce in yourself in the presence of a piece, is as valid as anybody else's, and they never get into conflict. And this is the nature of artistic knowledge. This is why it's so difficult to talk about it, to define it, to find any consensus, because it's not about consensus. It's the only field of knowledge where consensus is irrelevant and not necessary and not wished for in any way. And this is why we have pushed it outside of knowledge, because we always define knowledge as something that should be in some way consensual. even if it's temporary consensual. Well, there's one field of knowledge that does not care about that. Again, I'm not sure if this relates to to the second dimension there, but I do feel it's very important for people to understand how I process and how I make my artworks in collaboration with scientists, is that it's about working together and creating different things and understanding it in a different way. So the knowledge that I produce in myself as the artist is always different from the knowledge that's produced in, whether it's the scientist, the colleague of the scientist who engages with the art piece, or the person who comes from the street, whether they're really tiny or a well-known, I don't know, whatever. The knowledge that you create in yourself in the presence of an artwork is unique and immensely valuable. Mm-hmm. And it's that transformation that I was referring to when I was talking about this. Is again why I love this. Mm-hmm. This is why I wouldn't be a scientist, because I don't want to produce knowledge that's fact-based. I want to fight for the acknowledgement that the knowledge that's produced without consensus is absolutely fundamental for knowledge itself.
And what kind of reception does the kind of knowledge your practices producers have in the scientific community?
Well, the scientists would be able to tell you better what that is. I think the obviously it's refreshing. It's something that we do as humans. Scientists are humans too. So all of their knowledge is not only scientific based, and I think it is freeing in a way to understand that I suppose that in science the value of your knowledge is only valuable when you prove or disprove a specific thing, when you find a fact, when that fact is proven, or you discover something new that you contribute to the pool through the scientific method. But it's a very stressful and it's a lot of pressure to produce that knowledge and be relevant in your field of knowledge. In the arts, I think, I saw with my mother. My mother was a physicist and a chemistry teacher and mathematics teacher. All her life, all her career was that. When she retired, she decided to go into painting and learn painting. Are her artworks less valuable than mine? Of course not. Not in any way. And this is the freedom. This is why people say that art is therapeutic, because it allows you to value the knowledge that you're producing by making it or by witnessing or experiencing it without the need to argue about it, without the need for recognition in a way. Mm-hmm. You recognize the value of that piece, and it's not discussable. Mm-hmm. And I think this is why it's engaging in a way. This is why people want to go to an art exhibition. This is why they want to engage. This is also why the scientists want to engage with it, because it is very different in nature. And again, multiple perspectives are always enriching, and science will recognize that.
I would like to ask you about something else. I think to improve the collaboration between art and science, there's something that has to do with hospitality that is very important, to find a way to be together and recognize the other, and find ways that are rich for everyone involved. And I would like to ask you for an advice, something that you have learned along the way, something that you think that could improve in this field of art-science collaboration.
I think it's a field that is evolving. It's very different it was very different 25 years ago from what it is now. Right now, so I remember at the beginning of my career, every time I went into a research institute, and I was very privileged because I was in Oxford and I was studying in Oxford at the beginning of my career. But even when I left Oxford, every time I did a presentation or I started a residency, I would do a presentation about my work. And it was very important for the scientists to understand that I was genuinely interested in what they were doing, not just there to, because if it was secondary knowledge, then I could have gotten it from newspapers or secondary sources. I was really interested in what their research questions were and what they were trying to. So yes, it has that aspect. It's sort of interesting you say it as a hospitality. And most of the time we understand hospitality as from the perspective of the host, but actually hospitality is also about how you spend time in another person's place, right? Hospitality is not just about hosting someone, it's about how you behave when you're being hosted. And most of the time we forget that part. And so I suppose I did have very good experiences, and the butterfly laboratory was probably the most formative experience of mine. It was also the first. But I had friends who were scientists. I was already dating my husband when I went into the laboratory in Leiden to do the butterfly project. So I was taught to do my homework properly. Mm-hmm. And so to research about what they were doing in the lab before actually going there, so that my questions could be stupid, but at least they were relevant. Or they were based in ignorance, and ignorance is something that you can counteract. Stupidity is difficult to counteract. Again, you can't blame people for being ignorant. Why would you expect an artist to know about science, and why would you expect a scientist to know about the arts? So, this expression that there are no stupid questions, there are. Again, stupidity is difficult to counteract, but ignorance, it's easy to overcome. So ignorant questions are, I would say, very much allowed, or questions that are based on ignorance are very much allowed. Stupid questions are more complicated to deal with, and usually infer that you're not really trying to learn anything, just trying to be difficult. So yes, doing your homework will allow you to put more questions that are related to ignorance or lack of knowledge in a specific field, but that is fine to overcome, and that's usually respected. Come on, science is about this. And the formative experience that I had in Paul Breakfield's lab was doing a lab meeting in the first week that I was there. Nobody expected me to understand what they were doing, but I was encouraged to ask questions and to participate in the discussions in whatever way I felt was relevant, even coming from a field that had nothing to do with evolutionary biology. And when I presented my work, again, because I had this, I was expecting them to ask questions, not questions that would necessarily mean that they knew a lot about art, but that they really wanted to understand, and maybe this relates to that second dimension that you were talking about, which was, why do you say that you're making art here when I see you in the lab and what you're doing is exactly the same thing as we're doing? Yes. So, but that's not science. What you're doing in the lab is not science. Right. It's all the protocols that will give rise to the science. This is what I would say. So you're not, the scale is not really one-to-one, because if you're calling what you're doing on a daily basis in the lab science, then you're telling me that all the doctors that are not practicing doctors are no longer doctors. No, it's not. It's but this is the thing. Do you stop being a scientist once you stop working in the lab and doing the the experiments? No, you're still making science. The head of the lab is still making science. He's still making, or they are still very much knowledge makers in the scientific field. So it's not the protocols that define science, it's the questions that define science, is the way you pursue the questions that define science. So that would be the 1-to-1 scale. And maybe you're right. This is what I'm using. This is what I'm trying to tackle in a way. Is but I'm not producing science. The questions that I'm raising are in the forum of the arts. I'm not trying to find facts.
Great. Thank you, Marta.
I would say, since we have the coffee break now, would it be acceptable that instead of questions here, we can continue the conversation with the coffee? That applies also to you, Jaume, even if you are the boss here. So, where is the coffee happening? Upstairs? Okay. Marta, Lua, thank you so much. Thank you.